Okay, hello to all of you. Let's start discussing chapter number 11. And actually, with this chapter, we are starting the last unit. So in this unit, basically, we focus on genetics and evolution. So let me go into the slideshow mode. So in the last unit, we covered chapter number 11, 12, and then we have the last part is evolution, and it includes chapter number parts of chapter number 13, 14, and 15 from your book. So chapter number 11 and 12 is all about genetics. So if you read in your book, this story starts with the story of this girl. Her name is Emily, and she is the one who has a disease called cystic fibrosis. Okay, and um, and the important thing to know about this story is Emily's parents, they do not have cystic fibrosis. Okay, and I think in her family, her brother and sister also don't have cystic fibrosis. She's the only one who is the one who is suffering from this disease called cystic fibrosis. So first of all, we are going to talk about what is this disease, cystic fibrosis, and how this disease is inherited, and we are going to see how this is possible that her parents don't have the disease. How come she has this disease? So first of all, what is cystic fibrosis? Now, cystic fibrosis is a genetic disease, and this is a disease which is because of a mutation, and because of this mutation, there is a deletion of 3 nitrogenous bases, and as a result of that, so we have covered till now how transcription and translation works. So 3 nitrogenous bases, when they are removed in the mRNA, so in 3 nitrogenous bases they are removed. So eventually that information is transferred to mRNA, and then from mRNA a polypeptide chain is made. And so in the polypeptide chain, because of the deletion of 3 nitrogenous bases, there is 1 amino acid missing, and as a result of that, and this mutation what I'm talking about right now, it is in a gene called CFTR. So CFTR is cystic fibrosis transmembrane regulator. So this is a kind of a protein. So the people who suffer from this disease, they have a mutation in this gene. Okay, so in these people who suffer from this disease, they have a mutation in CFTR gene. There is a deletion of 3 nitrogenous bases, and as a result of that, in the protein, in the polypeptide chain, there is 1 amino acid missing. There is 1 amino acid missing, and as a result of that, the protein which is formed is not normal protein, and it is not able to perform a normal function. And as a result of the formation of an abnormal protein, because the protein is not normal, it cannot function properly. Because of that, these people suffer from a disease called cystic fibrosis. So I have posted some videos of cystic fibrosis also. Please watch them. Watch those videos. You will have a better understanding of this disease. So basically, in these people, their mucus is very thick. So it's a disease of lungs. So in these people, the mucus is very thick, and these people are very prone to the respiratory tract infections. So let's see how. As I said, it is because of a mutation, and it's a mutation in this kind of a gene. So now let's see basically what is the importance of this protein, and why this protein, when it gets mutated, it makes the people suffer from this disease. So let's come back here. So this CFTR, which is a cystic fibrosis transmembrane regular protein, the function of this protein is basically a membrane protein, and the function of this protein is in the transport of the chloride ions. Okay, the function of this protein is in the transport of the chloride ions across the membrane. Okay, so now in this case, this is the normal protein. Okay, this is a normal protein, and this is the protein. This is people. This is the cell people who are suffering from this disease. So you can see here, this is the functional CFTR. Ions can cross the membrane through CFTR protein. So this is the CFTR protein, and the chloride ions can pass through, and because these chloride ions can pass through this protein, then water can also flow in both directions. So what happens in these people, because these people, they have a mutation. So this is shown here, and this gene is on chromosome number 7. So this is shown here that this is your chromosome, this is the CFTR, this is a normal gene, okay, and this is the normal protein. But this is a mutated gene, okay, and as a result of that, these 3 nitrogenous bases, they are missing, and as a result of, there is one amino acid is missing, and as a result of that, the protein which is made, you can see this is a functional CFTR protein, this is a non-functional CFTR protein. So you can see how the shapes are different. So this is a mutant protein, okay. This mutant protein lacks a critical amino acid, causing it to have a different shape and different function. So this is a normal protein which has this shape, and this is a mutant protein and it has this shape. So what happens is, the people who have this abnormal protein, in these people the chloride ions cannot get transported through this protein. The chloride ions cannot get transported through the protein, and these chloride ions, they stay inside the cell. And as a result of that, what happens is the water moves inside the cell. Water moves inside the cell to dilute the concentration of chloride ions which is inside this cell. And as a result of that, what happens is, because the water moves inside the cell, outside the cell the mucus becomes very dry, and as a result of that, it becomes dehydrated, mucus becomes thick, and it can trap the bacteria and all that. And as a result of that, these people, they suffer from this disease called cystic fibrosis. So you can see here, this is the lungs of normal people who have the functional CFTR protein, and these are the lungs of the people who suffer from this disease. So basically, so I hope you understand what this disease is and why does this disease happens. Something very important I would like to tell you here is that, like, we are humans, okay? Humans are diploid organisms. So when we were having classes face to face, at that time also I have explained you that we all have, every species has a specific number of chromosomes. Human beings, we have 46 chromosomes, and these 46 chromosomes, they are present in 23 pairs. So 23 pairs, and in every pair, one chromosome is from the father, another chromosome is from the mother. So we have in total 46 chromosomes that define us as Homo sapiens, and we have 23 pairs. In every pair, one chromosome is coming from father, another chromosome is coming from the mother. So this is a karyotype, you can see. So you can see here, I think I have shown you this karyotype earlier also. You can see that there are 23 pairs of chromosomes. There are 23 pairs of chromosomes. This is chromosome pair number 1, 2, 3, 4, 5, 6, 7, 8, 9, up to 23. Okay. And the last pair is called the sex chromosomes. And the sex chromosomes, they can be XX if it is a female, or it can be XY if it is a male. So you can see here, so we have chromosome number, this is a chromosome pair number 7. So one chromosome is given to us by our father, another chromosome is given to us by our mother. So basically, because our chromosomes are present in pairs, which means that we have 2 copies for every gene. That's why we are diploid organisms. So we have 2 copies of every gene, and these 2 copies, they can also be called, they are the alternative version of the same gene, and they are called alleles. So we have 2 alleles for every gene. In the case of cystic fibrosis, you can see here, they're shown here. So this is chromosome number 7. So what is shown here is this chromosome, this is the chromosome pair number 7. So this is one chromosome, this is second chromosome, okay. So this is actually showing the chromosome pair of Amelie's parents, okay. So Amelie's parents, this is one chromosome, this is second chromosome. In both parents, one allele is normal, another allele is mutated, okay. So Amelie's parents, they are not suffering from this disease, they are normal. But if we look at their chromosome number 7, because chromosome number 7 carries the CFTR gene, one chromosome is having normal CFTR gene, other chromosome is having mutated CFTR gene, okay. So for this disease, if a person has one normal and one mutated, this person will be normal, because one of the allele of CFTR gene, if it is normal, it will be able to make the normal protein, and that much normal protein is able to perform normal functions. So a person to suffer from this disease has to has both alleles mutated, which means that for a person to have this disease, both alleles on the both chromosomes should be mutated, only then they are going to suffer from this disease. So in the case of Emily, her both alleles are mutated. This chromosome pair is from her parents. One is normal, one is mutated. So as a result of that, these people, one is normal, one is mutated, these people do not suffer from the disease, they are normal. But in the case of her parents, but because of her Emily herself has both alleles mutated. Both alleles are mutated. So just going over this terminology, as explained, what alleles are, what diploid organisms are, and what are homologous chromosomes. So you can see here, this is a karyotype showing all the 23 pairs. So every pair is called homologous because in this pair, whatever the gene is here, the same gene is here. So this is your paternal chromosome, this is your maternal chromosome. So whatever the gene, suppose here is a gene for skin color. So this is a skin color gene here on the paternal chromosome. The same skin color gene will be here on the maternal chromosome. So basically these pairs are called homologous pairs because they are quite similar in their length. They are also carrying the same kind of genes. So this already explained in diploid cell, one homologous chromosome is inherited from the mother, another is from the father. So this I already explained in cystic fibrosis. If one allele is normal, this person is going to be normal. It's not going to be diseased. Now, what is genotype and what is phenotype? So this is important terms for you to know. Genotype is basically the genetic makeup of an individual, and phenotype is the observable or the measurable feature of an. So genotype is the genetic makeup, which means how what kind of genes the individual has. Phenotype is the external appearance or the observable or the measurable feature of an individual. So basically, like if we look at the Emily's parents, one allele is normal, one is mutated. So which means they have one allele normal, one mutated. So their genotype is one mutated, one normal, but their phenotype is they do not have the disease, which means their phenotype, their external character is they are fine, they're normal people. Okay. So we know how genes are inherited. These things we already know. So basically, as you know that the genes, they are inherited to us by our parents. So how do our parents give us the genes? They give us the genes by the special kind of cells which we call gametes, which are the sperms in the men and eggs in the female. So basically, the genes are given to us by our parents through gametes, and the gametes of our two parents, they fuse by fertilization, and that determine what kind of genotype and phenotype we will have. Okay, so in the sexual reproduction, which is a normal way of reproduction, mother and father, they give gametes to their children. So the egg from the mother and the sperm from the father, they are formed by a process or a special kind of cell division which is called a meiosis. So by this cell division, so in the last chapter we have talked about a division called mitosis. So in which from one cell two cells are formed, and that kind of a cell division is taking place in our body cells, or we call them the somatic cells. We have another kind of cell division which is going on in our sex organs. That division is called meiosis, and by this cell division gametes are formed. And these gametes, they fertilize and they form new life. So it starts as a zygote and it then goes through different divisions and forms embryo, and then it forms a child. So this is the way through gametes, our parents, they give us the genes. So let's talk about this division. We need to know about this division called meiosis. It's a special kind of cell division by which the gametes are formed. So this division is different from mitosis, and it also has some similarities to mitosis. So meiosis, cell division, has two phases, meiosis 1 and meiosis 2. Meiosis 2 is exactly like mitosis. So I'm not going to go over I'm not going to repeat that. So meiosis 2 is exactly like mitosis, but meiosis 1 is different from mitosis. And meiosis 1 has 4 different stages. It's called prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay, let's go over here. Yeah. So this picture shows you this is meiosis 1, meiosis 2. Okay. So unlike mitosis, or if you go back and remember, review it, mitosis has 4 stages: prophase, metaphase, anaphase, and telophase, right? So these are the 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. Meiosis, it has 2 stages, meiosis 1 and meiosis 2. Meiosis 1 has 4 stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 2 has prophase 2, metaphase 2, anaphase 1, telophase 2. If you remember from mitosis, before the mitosis takes place, there is a phase called interphase. In the interphase, the cell is actually doing all the preparations. Cell is duplicating itself, it's duplicating all its organelles, it's making some proteins for the upcoming division. Similarly, before the meiosis can take place, there are some special cells in the sex organs. They undergo interphase. In the interphase, they are actually preparing. Okay. So after they have done the preparation, the cell enters into prophase 1, then metaphase 1, anaphase 1, telophase 1. So we are going to quickly talk about what happens in all these phases, and then meiosis 1 is followed by meiosis 2, and there is no interphase in between meiosis 1 and meiosis 2. So in the prophase 1, so basically there are all the other events are very same as mitosis, that this chromatin gets condensed and it takes the shape of chromosomes. The nuclear membrane starts getting fragmented, and the centrioles, they start moving apart because they have to form the mitotic spindle. So another important thing which is in the prophase 1 of meiosis 1 is that the pairing of these chromosomes takes place. So this chromatin definitely gets condensed. In mitosis, there is no pairing of chromosomes. In the prophase 1 of the meiosis 1, the pairing of chromosomes also takes place, which means that these chromosomes, they form pairs. So this pairing takes place. Okay, this pairing of chromosomes takes place in the prophase 1. Okay, and then in the metaphase 1, it is the pairs which come in the center. It's the pairs which come in the center, and in metaphase 1, these chromosomes, they separate. So if you remember what was happening in the metaphase of mitosis is all the 46 duplicated chromosomes, all the 46 duplicated chromosomes, they come in the center. But in the metaphase 1 of meiosis 1, it is the pairs which come in the center, and in the anaphase 1 of meiosis 1, it is the separation of the chromosomes. In the anaphase of mitosis, it is the separation of the sister chromatids. Okay, so in the mitosis, these chromatids, they get separate, but in the meiosis, anaphase 1 of meiosis, it is the chromosomes which separate. Okay, so that is why this division is also called reductional division. So 23 chromosomes go this side, 23 chromosomes go this side. So after meiosis 1, from one cell, 2 cells will be formed, and each cell will have 23 chromosomes. but remember that all these chromosomes are going to be duplicated. So these two cells which are formed after meiosis 1, then they are going to undergo meiosis 2, and meiosis 2 is exactly same as mitosis. So this cell has 23 chromosomes, this cell also has 23 chromosomes. In the metaphase 2 of the meiosis 2, the 23 chromosomes are going to come in the center, and here also 23 chromosomes are going to come in the center. And in the anaphase 2 of the meiosis 2, it is the separation of the sister chromatids. So this cell is going to form two more cells, this cell is also going to form two more cells, and here the separation is of the chromatids. So this cell will have 23 chromatids, this will also have 23 chromatids. Same thing here. So after one cell undergoes meiosis 1 and meiosis 2, there will be formation of 4 cells, and every cell will have 23 chromosomes. So we started with a cell with 46 chromosomes, we end up with 4 cells with 23 chromosomes, which is half the number of chromosomes. So the cells which have, so now our somatic cells, they have 46 chromosomes, which means they have 23 pairs of chromosomes. So the cells are diploid. But after meiosis, the cells, these are gametes. These are the sperms in the men and these are the eggs in the female. So these gametes which are formed, each gamete has 23 chromosomes. They have 23 chromosomes, and 23 chromosomes are half the number of chromosomes in a diploid cell. So in the diploid cell there are 46 chromosomes, now they have 23 chromosomes. So the gametes, they are haploid, and also the chromosomes, these cells, they are all the cells, the 4 cells which are formed, they are haploid, and all these cells which are formed, they are all unique because they have different set of genes. So there are some events which takes place during meiosis 1, which are responsible for the formation of the cells which are all different, all unique, and those 2 events which are responsible for genetic diversity, they are recombination and independent assortment. So I'm going to explain you what is recombination and what is independent assortment. So recombination is, so basically in the prophase 1, in the prophase 1 of the meiosis 1, so when the pairing of chromosomes takes place, so like you can see this is one pair, so these an event take place which is also called crossing over. So you can see here, this is like suppose this is the maternal chromosome, this is a paternal chromosome. These chromosomes, they exchange some of the genes with each other. Okay, so this is how these chromosomes are forming these kind of structures. This is called chiasmata formation or tetrad formation. You can see how they're crossing over. So this is the paternal chromosome, this is the maternal chromosome. So by this process which takes place during the prophase 1, crossing over takes place and they exchange some of the genes with each other. You can see this is the maternal chromosome. It has now some part from this chromosome, and this chromosome has some part from this chromosome. So this is called recombination, and this event is responsible for the formation of recombinant chromosomes. And because of the formation of recombinant chromosomes, the gametes which are going to be formed at the end of meiosis, they are going to be all unique. And the second event which takes place during metaphase 1 of meiosis 1 is called independent assortment. And in this process all these chromosome pairs, 23 chromosome pairs, they come in the center. 23 chromosome pairs, they come in the center. Now it's all a random chance. It's all a random chance that how these chromosomes they are aligned. Now suppose for the chromosome pair number 1, the maternal chromosome is on this side and the paternal chromosome is on this side. Now how the second chromosome pair is going to align, it is independent of the first pair. How the third chromosome pair aligns, it is independent of the second chromosome pair. Alignment means which chromosome goes on which side. So in this chromosome, like paternal is on this side, maternal is on this side. Now on this chromosome, it can be paternal is on this side, maternal is on this side, but it can be reverse also, and the same thing for this also. So this is independent assortment of chromosomes, and how these chromosomes they align, and how how they align is going to determine how they then so basically how these chromosome pairs they align is going to determine what kind of chromosomes are going to be going in the future gametes. Because if maternal chromosome is on this side, maternal is on this side, second pair is maternal on this side and paternal is on this side, then when in anaphase 1 these chromosomes they are going to separate. If paternal is on this side, for a second pair maternal is on this side, then this gamete which is going to be formed will have both the paternal chromosome for these pairs. But if it is reversed, which means maternal is on this side and paternal is on this side, then if that is the situation, then the gametes which will be formed will have paternal chromosome from this pair and maternal chromosome for this pair. So it all depends how they align. That is going to determine the kind of chromosomes the gametes are going to have. So how they align, it's going to determine what kind of chromosomes will be there in the gametes, and this is called independent assortment. So that is why the process of or the cell division called meiosis results in the formation of 4 gametes, and these 4 gametes they are haploid. Why they haploid? Because meiosis is a reductional division. It's a reductional division, and that results in the it's a reductional division, and that results in the reduction of the chromosome number. So from 46 chromosomes, the gametes which are formed, they have 23 chromosomes. They have haploid. Also the gametes of these two processes. Okay, so coming back, so now you understand the process of how gametes are formed by the process of meiosis. Now we're coming back to the same question of the cystic fibrosis. People with normal phenotypes can still pass CF allele to offspring, which was the case in the Amelie's story, that her parents are normal, but they can still pass CF allele to their children. People with CF will not necessarily have offspring with cystic fibrosis. It's also possible if a person himself or herself is suffering from the disease, they may not pass the disease to the children, because the gamete the person is passing to the children is going to be fused with the gamete from their partner, and if the partner is normal, or if the partner is giving the gamete which has a normal allele, then the child will not suffer from the disease, because this is a recessive disorder, which means that for this disease to occur, or this disease to show up, both the alleles they have to be mutant alleles. Both alleles they have to be mutated alleles. Only then both alleles if they are mutated, only then it is going to show up the disease. So cystic fibrosis is caused by a recessive allele, which means if for this disease to show up, both alleles they have to be mutated. An allele that affects phenotype only if the organism has 2 copies of that allele, that is a recessive allele. So a recessive allele is that allele that affects phenotype only if the organism has 2 copies of the allele, and it is hidden by a normal or dominant allele. So recessive alleles cannot express themselves in the presence of dominant alleles. So a recessive allele can express itself only if there is the absence of the dominant allele, which means that both the alleles they will be recessive. Only then it can show up. So recessive alleles usually they are shown by lowercase letters, and the dominant alleles they are shown by uppercase letters. So here is the case of Amelie's parents. So okay, before we go into this genetics, we need to know 2 more terms. What is heterozygous or heterozygote, and what is homozygous? So if both the alleles are identical, then that is a homozygote, or it's called a homozygous situation. So it can be homozygous dominant, which means both the alleles are dominant, or both the alleles are normal, so the phenotype is going to be normal. Homozygous recessive is when both the alleles are mutated alleles, recessive alleles. And in this situation, if both the alleles are mutated, the person is going to suffer from the disease. Phenotype is cystic fibrosis. Heterozygous is when the two alleles, one is normal, another is mutated. So in this case, the phenotype is going to be normal, but this individual is a carrier. So one is a normal allele, another is mutant allele. So now this is the story of her parents, Emily's parents. So this is the mother, this is the father. So both parents are heterozygous. Okay. So when they are heterozygous for cystic fibrosis, if the individual is heterozygous, which means one normal, one mutant allele, then this person is not going to have the disease, but that person is going to be a carrier. So that person can pass the disease to the children. So this is mother. So this is chromosome number 7. So one allele is normal, another is mutant. This is father, also heterozygous. One allele is normal, another is mutant. So during meiosis, by the cell division, so this is by meiosis, the father is going to make sperms and the mother is going to make the eggs. So as we just talked about, that these chromosomes, they separate. So this allele, which is normal, is going to be going in one sperm, because this is going to form one gamete, and the mutant allele is going to be going in the second sperm, because in the meiosis, in meiosis 1, these chromosomes, they are going to separate. And this is the case of the mother. So these are the two chromosomes. One allele is normal, another is mutated. So in meiosis, these chromosomes are going to separate, and it results in the formation of two gametes. So one gamete will have normal allele, another allele will have mutant allele. So now let's see, these are the two kinds of eggs and these are the two kinds of sperms. So when this sperm fertilizes this egg, the child who is going to be formed will be having two normal alleles. So this child is going to be unaffected, and this child is not going to be having any disease. Now for when this sperm fertilizes this egg, the genotype, okay, so in this child, AA is the genotype, which means both the alleles are normal, and this child is, its phenotype is also normal, which means no disease. In this case, when this is the egg with the normal allele, this is the sperm with the mutant allele. When this sperm with mutant allele fertilizes this egg, the child who is going to be formed, its genotype will be capital A and small a. So this child is going to be carrier, but its phenotype is going to be normal. And the same case is happening here. If this sperm fertilizes this egg, the genotype is going to be capital A and small a. So this child is going to be carrier, but its phenotype is going to be normal. And the same case here, if this sperm fertilizes this egg, now this child, its genotype is both the alleles, they are mutant, and this child is going to suffer from the disease. So this is its phenotype. Okay, its genotype is both alleles are mutant, and this child is going to be suffering from the disease. This child will have cystic fibrosis. So this is what is the situation of Emily. So that's all this chapter. So basically, this is the way we can predict the future, the way we can determine the future progeny. We can do like this, and this diagram is called the Punnett scale. So Punnett scale is a diagram to determine the probability of offspring having particular genotypes. Okay, this is called Punnett scale. So it is based on the genotypes of the parents, and it matches up all the possible gametes. That's the way we did that. This mother has these two gametes, father has these two gametes. If this fertilizes this, what kind of genotype and phenotype the child will have. This is what is called the Punnett scale. So that's all in this chapter. If you have any question, please let me know.